All right, so today we're going to be talking about the molecular structure and organization of chromosomes. This is very interesting work. This wasn't able to be completed until like the second half of the 20th century. Mostly in the 70s and 80s, really did we get a good picture of what chromosomes look like, and we weren't able to really manipulate chromosomes to get to some of the finer detail of chromosomal structure until the 80s, basically. And so this work that you were going to be looking at, work that's done in bacteria and in eukaryotic cells, but we're going to skip organization and structure of chromosome in viruses, all right? So let's get going. So chromosomes are structures that contain genetic information. We know that, right? They are what we call complexes of DNA and proteins. What does that mean? A complex is a large molecule made of proteins and something else. So remember, you've heard the word chromatin. That equals DNA. Sorry, guys, I'm going to try to write a little more slowly. Maybe this will work. Plus its associated proteins. Oh, there we go. Let me just erase that, and it'll go back to its original size, hopefully. Maybe not. So, complexes of DNA and proteins. We're going to get to that in a little bit. The genome itself consists of genetic material, all the genetic material that an organism has. We know that in bacteria that the chromosomes are circular, and in eukaryotes we know that they are linear and that we have a complete set of what we call nuclear chromosomes, all of our chromosomes that have our genes, but we also have mitochondrial genomes, which are circular, and plants have chloroplastic genomes, which are also circular. So remember those genomes when we're talking about cell division. Now, the main function of our genetic material is to store information, right? So DNA molecules have certain jobs to do. What are those jobs? They make RNA, they synthesize or make RNA. You know, I am getting about done with how this is working. I want my pen. I don't want, capture cursor while drawing. Sorry, guys. Synthesis of RNA. We have the replication of chromosomes, the proper segregation of chromosomes during M phase of the cell cycle, and then the compaction of chromosomes in cells so that our cells can have all the DNA they live, but within the nucleus. So remember, all of our chromosomes have to be packed into a little tiny, tiny space of a single cell that you can't even see with the naked eye, right? So that's a big job, compaction. So bacterial chromosomes, how do they accomplish this? Bacterial chromosomes are much smaller than eukaryotic chromosomes. In E. coli, they have about 4.6 million base pairs. Influenzae, 1.8 million base pairs. That's dinky compared to what eukaryotes have. Typically, what you have are sequences that are structural genes. Structural genes are the genes that are used to make the proteins that allow for the organism to survive. So whenever you see structural gene, that means that that gene is an important gene. It does something. You have non-transcribed regions between those structural genes. Those are called intronic regions. So bacterial chromosomes don't have a whole lot of these. They have a few, but not many. They have mostly structural sequence. So this is an example of a circular chromosome, right? We've seen circular chromosomes before. And let's look at a few details. Right here we have what's called the origin of replication. The origin of replication is where replication starts in bacterial cells. We've talked about that before. Each one of these regions here are intronic regions, okay? What the introns are important for is, and they're the red zones here. If you get real close, you can see that. They're really important because, well, they're not intronic, they're called intergenic, they're kind of intronic. But what that means is that they're used for packaging. We'll look at how that packaging happens and folding in a few minutes. And then we have these little zigzag regions here. These are called repetitive sequences. Repetitive sequence you'll find throughout all chromosomes, and we're not entirely sure always what those repetitive sequences mean, but they do serve a special job in packaging chromosomes because if you get rid of them, the chromosome no longer folds appropriately. So they are important. So The structure of bacterial chromosomes, remember these guys don't live inside of a nucleus, but they do live in a special spot called the nucleoid region. Nucleoid is not surrounded by a membrane, it's just DNA floating in the cytoplasm. So how do we keep this really important molecule here? This giant donut, right? Can you tell it's morning? I always think of donuts in the morning. This giant molecule organized. Think about it like a necklace, right? You try to keep your necklaces, and the ladies might be able to, or men too, relate with this. You don't throw your necklaces around because they become tangled, right? DNA is the same way. It's floating around inside of the cytoplasm. So if you don't organize it, it's going to become tangled, and the chromosomes may not be able to appropriately express genes if they're all tangled up. Also, to fit within the bacterial cell, you have to compact this thing about 1,000-fold. So in order to do that, bacterial chromosomes have developed this mechanism of folding into what we call looped domains. And there are DNA binding proteins that bind to specific regions on the chromosome. Wonder where those could be. Let's go back to that. Where do you think DNA binding proteins would bind here so that they can all come to the center and form this flower-shaped looped chromosomal structure? Think about it. The number of loops is going to vary based on the size of the bacterial chromosome, which makes sense. Bigger chromosome, more petals on the flower. So we call this looped chromosomal DNA. On top of that, there is a phenomenon called DNA supercoiling. DNA supercoiling further compacts DNA into a smaller structure. So supercoiling happens when you have these loops introduced into your flower petals. So first you have DNA binding proteins associated with certain regions of the chromosome, and then forming these big loop domains, and then the loops are supercoiled. So how does supercoiling take place? Well, we have what the schematic is showing you is a circular chromosome laid out flat. These plates on the end are going to prevent unraveling. And so what happens is basically the chromosome starts being turned so that these loops kind of bubble out. Now, I'm going to ask you guys to imagine something that maybe you might have seen in the old days. So in the past, telephones used to have long telephone cords. I really wish I could show you this. I don't have one with me, unfortunately. Telephone cords actually have this alpha helix structure. Now, my grandmother had one telephone in her house. I'm old, guys. And her telephone cable stretched all the way across the kitchen. It was a really long cable. It probably would stretch 20 feet if you went all the way into the other room. She used to walk around talking to people using her handset, which attached to the telephone. And as she would walk around, she'd turn. And what that would do is it would coil up the telephone cable. As the telephone cable got turned and turned and turned, these supercoils would evolve out of the telephone cable. And so in the same way that a telephone cable can be overwound or underwound, DNA does the same thing. DNA, what will happen is if you take turns out, if you add fewer turns, if you uncoil it, you'll have a coil that goes in one direction, all right? If you supercoil it, you tighten the coils, so here you're going like this way to the left. This way, if you add more turns, which is typically what my grandma did, you would end up with coils coming out on the side. So what you're doing is you're tightening up the coil, and that introduces more turns in the double helix. It kind of bubbles out like this. So basically, each of these conformations can be introduced by a class of molecules called topoisomerases. Topoisomerases are going to either undercoil and produce a loop on the right side, or supercoil and produce a loop on the left side. So supercoiling is going to influence the function of chromosomes, all right, big time. So if you have one negative supercoil per 40 turns in the double helix, negative supercoiling is going to help compact the chromosome. It creates tension, though, within the molecule, which causes strand separation to happen here. What does that mean? If you have a negatively supercoiled region here, And they're showing it with the chromosome laid out. Think of this within one of the petals of the flower that we looked at a while ago. That actually causes this to be looser, and it allows DNA replication to happen. And this is exactly what happens in the origin of replication. You have negative supercoiling that induces an opening of the double helix because it puts tension on the hydrogen bonds between both strands, and boop, the strands open up, and that allows enzymes to interact with the origin of replication. We'll be seeing that in more detail in a few chapters. So, what's doing the supercoiling? DNA gyrases, or DNA gyrase in general, is a molecule that falls within the type 2 topoisomerases, or DNA topoisomerase 2. It induces negative supercoiling. It definitely uses ATP energy. So this is an active process. You need energy for supercoiling to happen. It can also relax certain positive supercoils. When that happens, when you have relaxation occurring, genes within that region can be expressed. Now, DNA topoisomerase 1 also relaxes negative supercoils. So topoisomerase 1 and 2 do two different jobs. You don't need to worry about that right now. Specifically, like what types of genes they open up or if it's the origin. You just need to know that topoisomerases relax and induce negative supercoiling, all right? And so what this does is it allows supercoiling and relaxation to occur for transcription. How does this work? Well, the topoisomerase molecule, both of them, have what's called subunits. 2 subunits on the bottom, or the A subunits, and 2 subunits on the top. The DNA molecule strings through the A subunits, and then it knots back around through the B subunits, forming, and what happens is you have this clamp on the DNA, and this clamp allows DNA to form almost like a single figure. Well, it's a kind of a figure 8 if you look at how it works here. And so topoisomerases clamped onto DNA in numerous regions are going to form these 2 negative supercoils. How cool is that, right? This looks like a sailor's knot, if you ask me. So gyrase induces negative supercoils, like we just talked about, because it's a topoisomerase, right? And without it, bacteria are not going to survive. Blocking topoisomerases actually kills bacteria. And so 2 examples of medications that block topoisomerases are the quinolones and the coumarins. The quinolones and the coumarins are both antibiotics. They don't inhibit eukaryotic topoisomerases. So when somebody takes ciprofloxacin, Cipro, you guys have probably heard of that. Ciprofloxacin is a quinolone, and it's a really strong antibiotic. It can kill anthrax, you guys, and other very serious bacterial diseases. I think that it also can get rid of certain strains of streptococcus. And so what happens is when you take ciprofloxacin is Cipro stops topoisomerase activity, basically gene regulation, chromosomal regulation in bacteria, but it doesn't interact with your own personal eukaryotic topoisomerases or gyrase. So it kills preferentially the bacteria, but not your proteins. So when somebody has an infection, they can take Cipro and be just fine. How about eukaryotic chromosomes? How are these things regulated? Well, we know that we have linear chromosomes. We have, how many pairs? I'll let you guys remember that. They're all regulated by supercoiling as well. The total amount of DNA in a eukaryotic species is typically greater than bacterial cells, so that means we have to package them even tighter. Remember, they all need to live in the same nucleus. So let's talk about organization of eukaryotic chromosomes. What do we have that are characteristic appearance markers in a strand of DNA? There are origins of replication, just like there are in bacterial chromosomes. Bacteria have one, eukaryotic chromosomes have many. Centromeres. Prokaryotic chromosomes do not have centromeres, but we do. That's that little center skinny waist. And then telomeres. Eukaryotic chromosomes have end caps called telomeres that protect the chromosomes from fraying. Bacterial chromosomes, since they're circular, they don't require telomeres. Just a few details. Several origins of replication per eukaryotic chromosome. Centromeres are condensed regions of the chromosome which help the chromosome segregate. Sorry. Quick fact, Dr. Val gets up at 5 a.m. to record these, so I yawn sometimes. Sorry. Centromeres allow the chromosome to segregate during mitosis and meiosis because the centromeres contain the kinetochore, right? Remember that? And then the telomere prevents translocations actually, but also helps maintain chromosomal length. So here's a schematic showing each of these details. So here are several origins of replication localized on this schematic of a chromosome. Here is the centromere, which is required for separation or segregation during meiosis and mitosis. This is what those spindle microtubules attach to pull sister chromatid A from sister chromatid B. And then the telomere down here and up here prevent translocations, basically inappropriate swappings of DNA between two chromosomes during meiosis, but it also prevents the chromosome from getting shorter every round of mitosis. There is a certain point at which telomeres do shorten. Old DNA is shorter than new DNA. So people with, if you compare the DNA chromosomal length between a 90-year-old and a 9-year-old, you will see length differences. Now, every chromosome has at least several hundred genes on it, up to several thousand. It just depends on the length of the chromosome. Genes are going to be located between centromeres and telomeres. And in lower eukaryotes, and when we say lower, these are simple organisms like yeast, maybe blue-green algae. The genes are pretty small because those genes are still, we consider, very primitive. Very few introns. So primitive genomes, you don't see a lot of intronic DNA. Now in higher eukaryotes, mammals, vertebrates in general, genes are very long. They tend to have many introns, and those introns have variable length from 100 base pairs to 10,000 base pairs. So the size of eukaryotic genomes vary based on actually a whole lot of intronic sequence. So I'll show you that in a few minutes, the relative sizes of eukaryotic genomes. It turns out that the size of the genome does not predict the complexity of the organism, and you'll see that in a minute. In some cases, the variation is due to the complexity of the species. So you can look at two really closely related salamander species, and one will have a small genome, the other one's really big. The difference in the size of the genomes isn't usually, again, like I was saying, because of extra genes. It's because of those intronic or what we call repetitive DNA sequences that don't code for anything. So here's an example of two really closely related salamander species. Okay, salamanders are organisms that live near water. They look kind of like lizards, but they can't live on dry land. They need to be around water. They have real thin skin. And if you look at the genome between the Richmondii and the Larcelli species, the Larcelli has more than twice the amount of DNA that the Richmondii does. And that's just because of continuous repetitive sequences. So what are those? So repetitive sequences are basically sequences that have lots of A's and T's in them. So sequence complexity refers to how many times you have a base pair sequence occurring in the genome, A, T, G, C, right? We have unique, non-repetitive, those would be genes. Moderately repetitive, some of those are what we call DNA binding regions. We'll see that when we talk about gene regulation. And then you have these highly repetitive sequences. So first, let's start with the unique non-repetitive. You find it once or twice in a genome. These are definitely structural genes. In fact, these would be alleles. That does include introns, other non-coding DNA inside of eukaryotes. But basically, you're looking at genes, and this makes up about 41% of human genomes. So here are the regions that encode for in the human genome. The four classes, regions of genes that code for proteins, only 2%, only 2% of our genome is made up of exons. Introns and other they are important regions of genes, make up 24%. Unique non-coding DNA, these are usually regulatory regions, which we'll get to later. 59% of our genome is made up of repetitive DNA, stuff that doesn't code for or regulate gene sequences. So, only about 40% of our genome is responsible for expressing who and what we are. And that's pretty astonishing if you think about it. So now the repetitive sequences, we break those down between moderately repetitive, few hundred, few several thousand times. These are genes that make ribosomal RNA, genes that make histones as well, which we'll get to in just a little bit, origins of replication, and transposable elements. We'll talk about those in chapter 21. Those are really cool. Now, the highly repetitive elements, you find these tens, thousands of times in an individual chromosome. They're pretty short, several nucleotides. Here's an example, AATAT or AAT. These are repetitive sequences that you'll find sprinkled throughout the Drosophila genome. In humans, we have something called the ALU family of repetitive sequences. The ALU family is just A's and T's, lots of them sprinkled about. There's one that is very characteristic in humans. In fact, we tend to do a lab in Bio 4A and in biotechnology to identify who has the AT repetitive ALU element, and we can actually look at how many copies you have, 1 or 2 or 0. That's transmitted through families, but it has nothing to do with an organism's or an individual's phenotype. You typically find these near centromeric regions. They don't regulate the centromere, they're just there. So let's look at chromatin compaction in eukaryotes, all right? We looked at chromatin compaction or chromosome compaction in bacteria. Remember the little flower shape and DNA supercoiling? Well, chromatin in eukaryotes still goes back to, remember, DNA plus its associated proteins. That equals chromatin. Okay, don't be surprised if that shows up on an exam, all right? Chromatin, we talk about that a lot when we look at, the model of the cell. Oh, that's chromatin. It's not just DNA, folks, all right? So if you stretch out a single chromosome or a single pair of chromosomes, you'll see that when you lay them end to end, it's over 1 meter long. But a cell's nucleus is only 2 to 4 microns long, okay? Remember, 1 micron is 1/1000th of a millimeter, and you know how dinky that is, right? So DNA has to be coiled up super, super tight so that we can fit 46 of those puppies inside of a nucleus that you can't see with the human eye. And it still has to be loose enough so that genes can be expressed. So compaction of long linear eukaryotic chromosomes involves play between DNA and histones that we're going to call, or proteins that we call histones, okay? So DNA protein complexes, chromatin, include histone proteins plus DNA, and changes in their organization result in chromatin compaction. So histone proteins come in a few different flavors, all right? We have the H2A, H2B, H3, and H4. When the histones interact with and form what we call a histone octamer, DNA is able to wrap around the octamer like string around a spool of thread, or the thread wrapping around the spool. We call a single loop of thread. So let's pretend I have a spool here. Oops, I have to turn on my pen. Let's pretend I have a spool here, and let's change our color for DNA, which will be blue, or I don't want gray, I want blue. All right. DNA is going to wrap around this histone octamer one time, and this is what we call a nucleosome. I'm just going to write it down here, okay? So the nucleosome consists of our histone and our DNA wrapped around it. And the histone itself, remember, is made up of 4 molecules, 2 repeating. So you have H2A, H2B, So we call the histone an octamer, and then there's another one that clamps the DNA in place. So the nucleosome is a single spool, or you could think of it as a bead, and the DNA wrapping around it once as a piece of string, okay? So we call this nucleosome structure, when you have repeating nucleosomes, the bead on a string, all right? So let me get to that. It turns out that you end up with 1.65 negative superhelical turns around the single octamer. Here is a better representation of what I'm actually talking about. So H4 happens twice, H3 happens twice, H2B, and then H2A is the clamp, all right? So DNA wraps around actually 1.5 times. It just looks weird. I couldn't do 1.5 times because I'm not that good of an artist, guys, sorry. So here's DNA wrapping around the histone octamer. Here's what we call the linker region. It's just a part of the DNA strand. And then one of the things I want you to pay attention to is this amino terminal tail. This little tail helps lock DNA onto the histone octamer, okay? And so depending on whether or not these tails are locking DNA down, or like in this example, or if the tails are loose and not locking DNA down, that actually helps regulate whether or not genes in this region are turned off or turned on. We will talk about that when we talk about gene regulation, and I have a really kind of interesting, crazy story to tell you about that. And it's a personal story, so I won't get into trouble. I won't sue myself over my own story, right? So this is what we call the beads on a string theory. Short structures of histones with DNA wrapped around it. So histone proteins are basic, it turns out. And when I say basic, that means they have a charge. They have positively charged amino acids, lots of them, lysine and arginine, and they bind to the negatively charged phosphates on DNA. Remember, negative and positive molecules attract each other. And so we have these electrostatic interactions and hydrogen bonding that help keep DNA bonded to the histone octamer. Histone proteins are have that globular domain and that tail domain that helps keep things kind of glued together. We already talked about the histone types, H2A, H2B, H3, H4, and then H1, I said H2A, I apologize, back here. H2A bubbles it out. H1 is the clamp that holds the histone onto the DNA. It's a binder DNA. Okay, non-histone proteins also affect this. That's a different regulatory mechanism. We'll talk about that later. So here's an example of the coils around histone. Here's histone H1. They're just showing the histone octamer here as one clump. Here are those amino domains that are going to lock down on the DNA molecule or not. And then here are the non-histone proteins studying the linker DNA, keeping it open and available for interaction with other proteins. So the structure of the nucleosome was originally proposed in 1974, remember, 70s and 80s, by Roger Kornberg. Kornberg based his proposal on a series of really kind of fun experiments. He used biochemical experiments where he had enzymes available to kind of chop the beads off the string. He used X-ray diffraction studies so that he could look at the structure of the histone molecules. And then he did electron microscopy so he could actually see he could basically take an X-ray. But not diffraction, a different type of an X-ray of what the molecule looked like when the beads and the string were put together or when they were melted apart. Now, Marcus Noel came along a little bit later, in the later 70s, early 80s, and he tested Kornberg's model with more detail. And what he was looking for was digesting the linker region with an enzyme that should give you fragments that are really short, about 200 base pairs long, because they hypothesized that the nucleosomes would have pieces of DNA on it that were about 200 base pairs long. And so the idea was that linker DNA was more accessible than core DNA or DNA wrapped around a histone. So DNase 1 would melt this region right here. And I shouldn't say melt. It literally goes in and cuts the DNA. So when you looked at it, you would end up with digested pieces of DNA. So here's the experiment. So in the experiment, you take DNase 1 and you incubate DNA with DNase 1 in different concentrations, all right? You have low concentrations of DNase 1, medium, or high. Let me change my color here so we can see it a little better. All right, you allow incubation to occur for a period of time, and then you treat your mixture with detergent and phenol. What the detergent does is it basically stops the reaction, and the phenol allows you to separate DNA from other components, okay? You then purify your DNA. So here's the DNA before digestion, or at least the conceptual level, a cartoon of what's happening. So there are your histones, the beads on a string. You're going to be digesting the string so that you should have one or maybe two beads on the string using the DNase 1. Okay, you stop the digestion. You remove the histones using the detergent and the phenol, and then you run your DNA down a gel. The marker is just something that tells you how big the pieces of DNA are. And after you run the gel, you stain it and you look at it under UV light. The stain allows you to see the size of DNA fragments. And this is what they saw. In the low concentration of DNase 1, okay, the low concentration of the enzyme, you saw many different fragment sizes. Here's one, here's one, here's one, here's one, here's one. But they always happened at repeating units of 200. At the medium concentration, you saw the 200 base pair and 400 base pair, but nothing larger than that. And then at the high concentration, you only saw 200 base pair. What this means is that you had full digestion of all linker DNA, and all that you had was 200 base pair fragments that run down the gel. So in case you don't know, when you run DNA down a gel, the large pieces sort to the top. So this is big pieces of DNA, and this is going to be smaller pieces. This is an example of like being small. You know, you can get through something real fast. When you're big, you can't get through something real fast. It's like a bouncy house. Well, not a bouncy house, but maybe like a ball pit or something. And so the smaller molecules get through the matrix of the gel faster than the large ones do. So these were the data that demonstrated that the nucleosome structure was about 200 contained about 200 base pairs worth of DNA surrounding the repeating histones. So here's an interpretation. At low concentration, DNase 1 didn't cut the linker entirely. So you have different size fragments. At high concentration, you only have the 200 base pair fragment because, now let's do it this way. If you have a long piece of DNA, all right, and it's cut every 200 base pairs, all right, depending on how much enzyme you add, it's going to result in cutting either very consistently. So if you have enough enzyme, you get all the cuts complete. But if you don't have enough, you don't get all the cuts. Therefore, you end up with this pattern here with only moderately cut DNA. If you have questions about that, shoot me an email. Nucleosomes join to form a fiber, all right? These fibers exist to help package and compact DNA. So histone H1 helps compact by holding the DNA onto the histones, hence holding the beads onto the string. And if you prepare DNA, intact DNA, with different amounts of salt, you can actually look at compaction either in moderate salt concentrations. You'll see the classic normal bead on a string. When we say moderate, that's about the amount of salt in a normal cell. If you change the salt concentration, you wash the salt out, you'll see more compaction. So you have to have the right amount of salt around for compaction to occur. What are salts? Positively charged ions. What are the histones in DNA? Histones and DNA. Remember, histones are positively charged. DNA is negatively charged. You get rid of the salt, that changes the overall charge in the solution, which changes the compaction pattern. So the 30 nanometer fiber shortens 7-fold. The structure then can become a more compacted model, and that gives us our solenoid model here. The solenoid model is supercompacted DNA. Okay, so here would be core histone molecules with the DNA wrapping around it. Here you have the zigzag model. This is a little unusual. However, the cool thing about this is that you would have availability for these regions for transcriptional activation, right? So this is the difference between the solenoid model and the zigzag model. So the third level of compaction occurs at the nuclear matrix model, or there's a model called the nuclear matrix, right? This is where you have the nuclear lamina, fibers inside of the nuclear membrane, contacting pieces of the loop domains. So here's the nuclear envelope, okay, the outer nuclear membrane. Remember, it's a double membrane. Inside you have proteins associating with DNA, with our chromatin, and then here's the nuclear lamina, these regions right here, all right, in kind of the flesh color. See if I can get my pen going here. Here's the nuclear lamina right there. So interactions between DNA, histone proteins, and the nuclear lamina kind of help hold chromosomes in certain regions. Now, why do you not see that here? Remember that the nucleus is three-dimensional. You're only seeing a two-dimensional representation of what's happening inside of the nucleus in this picture. Those regions might be touching the far wall of the nucleus or closer to you if you were to be looking at this three-dimensionally. So radial loop domains, what are these? These are really cool. These are called Mars and SARS regions, and basically these are the regions that are going to anchor DNA to the nuclear matrix, forming those little loops. So here's a matrix attachment region. Okay, we call them the MAR, right? MARs are anchored to the nuclear matrix. So right here's a protein fiber, here's a Mars region, here is a piece of the chromosome. What's cool about this is you have the protein matrix allowing regions to be available for transcriptional activity. All right, so why were these radial loops so exciting when they were discovered? Well, first of all, it helps control what genes are transcribed, and it kind of helps control the rate as well. It also allows us to predict where in the nucleus you're going to find a chromosome, and it helps support the chromosomal theory of inheritance, right? Because each chromosome in the nucleus is located in a specific non-overlapping territory, right? So it helps support the chromosomal theory of inheritance because they're your chromosomes and they're available for transcription. So this leads us to a discussion with the differences between heterochromatin and euchromatin. Heterochromatin and euchromatin are regions of DNA that are either super tightly compacted, where genes cannot be transcribed, or they're very loosely compacted. Euchromatin is less condensed than heterochromatin. Euchromatin, when you look at DNA, are going to look kind of like fluffy regions of DNA. They're really obvious in, believe it or not, Drosophila salivary gland DNA. And people really do pick out Drosophila fly salivary glands to look at DNA. These euchromatin regions are extremely transcriptionally active. The loops are much more open. Therefore, when you look at the chromosomes, those regions look fluffy. They used to call them polytene puffs, if you've ever heard that term, because those are the polytene chromosomes in the salivary cells in Drosophila. Now, heterochromatin are tightly compacted regions. No genes are transcribed here. They're very, they have like tight compaction. Those radial domains are super coiled even tighter. So if you stain these chromosomes, heterochromatin looks really dark and euchromatin looks really light in comparison. What is happening? So we have inside of heterochromatin regions that are always going to be transcriptionally active. They're permanently inactive with regard to transcription. I said that they were active, they're inactive. We're talking about heterochromatin. For some reason, I usually like to think about euchromatin first, but in this set of slides, the heterochromatin is coming in first. So heterochromatin is always inactive. There is facultative heterochromatin where you have a region, say for example, like a bar body, that is heterochromatin, but it gets fluffy during mitosis. So you have a reversal of heterochromatin to euchromatin, then back to heterochromatin again. So here's an example of euchromatin, nice and loose, and then heterochromatin near the centromere, near the telomeres, very, very tightly compacted. If you were to put a stain or a dye on this, let's say a red one, this would look pink and these would look dark red. And that's how they were characterized in the polytene puffs and polytene chromosomes of salivary glands in Drosophila. So if you think of the cell cycle, in metaphase, remember this is where cells are in M phase. In metaphase, what's happening? The metaphase plate, the chromosomes all align on the metaphase plate. So cells that enter M phase have a level of compaction that is pretty extreme. Basically, sister chromatids all migrate to the center metaphase plate in preparation for separation, right? Sister chromatids at that point are entirely heterochromatic. In other words, there are no genes being expressed. There shouldn't be genes being expressed. The cell's getting ready to divide, right? Two parallel chromatids have an overall diameter together of about 1400 nanometers. Now, it is a little different depending on chromosome number, right, in humans, because different chromosomes are different lengths, but that's just a general estimate. So highly, highly condensed metaphase chromosomes basically are transcriptionally inactive. Those radial loops are really highly compacted, and if you think back to those proteins on the inside of the cell membrane on the inside of the nuclear membrane, sorry about that, we know that those proteins allow the release of histones so that the chromosomes can travel to the metaphase plate. So you have a release of that structure as well. Histones are needed for that compaction. So here's an example of the DNA double helix. Here are the histones, remember beads on a string. And then here is a 30 nanometer fiber of these repeating histone, the zigzag model here of the repeating histones. In addition to that, we have these protein scaffolds, all right? These are called packaging proteins, DNA packaging proteins, or scaffolding proteins, through which the beads on a string zigzag. And then this is where these scaffolding proteins zigzag themselves to make a super tight piece of heterochromatin. So I think one of the things that we can take away from these examples is supercoils, alpha helices, and these zigzags are a way that nature in general packages and stores very large complex molecules, right? Carbohydrates are a different deal. However, they even follow a helical format if you look at large enough carbohydrates. So this is a really interesting topic or shape for biological function. So here's a picture of what DNA looks like when you remove DNA itself from scaffolding proteins. So this is an example of a metaphase chromosome. I don't know which chromosome, but if you take a metaphase chromosome, normal metaphase chromosome, and you dissociate it from the scaffolding proteins using salt, because you can do that, salt will remove the histone proteins. Here's what the DNA looks like, just DNA dissociated, the DNA strands. And here's what the scaffolding proteins look like. And the scaffold, look at that, looks just like a pair of sister chromatids. So you could think about this like the tree trunks and the leaves on the tree. So multi-protein complexes are going to play a role in metaphase chromosomes. Condensin and cohesin. Condensin helps the chromosomes condense or coil up, and cohesin play a role in when the sister chromatids align with one another. So these are called SMC proteins, structural maintenance of chromosomes proteins, and they require, again, ATP to change the structure of a chromosome. So that's an active process. So here's an example of one of these proteins. They have certain regions, a head region where ATP can bind right here. We have N and C terminals, and then here's a hinge region. It's pretty big, 50 nanometers. So condensin starts working as M phase begins. Remember, you have to condense these chromosomes up. They help compact the sister chromatids, bringing them together. Well, not bringing them together, they bring together homologous chromosomes. But they bind to the chromatids during compaction. How it wraps around at the time that this lecture, well, actually, we still don't exactly know how that works. But it seems that without condensin, the chromatids don't organize and condense appropriately. So during interphase, so here's condensin, it starts out actually in the cytoplasm. So during interphase, during G1, S, and G2, there's a signal that causes condensin to go into the nuclear space. The theory behind that is that if condensin gets in there without, just kind of randomly, the chromosomes will start condensing. So you have to keep them separated, right? So you have euchromatin occurring for transcription during normal, non-cell cycle activities during G1. But during S and G2, that condensin at the end of G1, condensin starts to enter the nucleus so that once the chromosomes are duplicated, heterochromatin is formed, and you end up with those loops. So cohesin then becomes important because that's going to bring sister chromatids together in meiosis, all right? After S phase, during the middle of prophase, sister chromatids remain attached along their length, and it can help promote homologous chromosomes to come together. And in certain species, the arms of the sister chromatids separate for certain processes to happen. You don't need to worry about that. Here's the upshot. You have a pair of sister chromatids. Cohesin holds the chromatids together. You begin condensation. And then at the end of prophase, cohesin only holds on to the centromere region, okay? The other cohesin molecules are released. And then at the end of M phase, going into anaphase, those cohesin molecules that were associated with the centromere and the kinetochore release, allowing the separation of sister chromatids. Now, again, remember, cohesin is going to be important during meiosis for pairs of sister chromatids or homologous chromatids to come together. So you'd have a separate set of cohesin molecules right here as well, and the second pair of sister chromatids. So imagine this is sister chromatid pair 1 of mom or dad, maybe, I don't know. You'd have the separate sister chromatid 1 from the other parent here, and they're held together by cohesin. So you would have cohesin, and they're slightly different versions of cohesin. Don't worry about that right now. But you would have the first cohesin release during M phase 1 and the second cohesin release during M phase 2 of meiosis. All right, that's the end of this chapter. Remember, if you have any questions, send me an email, and I'll catch up with you on chapter 13.